Hello, everybody. Thank you for bearing with us, and I'm glad everyone was able to get in with the new Zoom link. Thank you so much for attending the Bee Conservancy's BTV webinar today on pesticides, bees, and the race to curb neonics. Just a few housekeeping rules to get us started as the last few folks get inside. So to keep things running smoothly, we invite you to change your name, add your pronouns and organization if you'd like. Everyone is muted by default, but you can throw your location in the chat where you're coming from. And the chat will close after the presentations start, but you can continue to drop your questions in the Q&A and upvote your favorite questions. We're so excited that you're with us today, and if you're interested in more unbelievable programming, consider joining us for World Bee Day if you're in the New York City area on May 21st. You can register at thebeeconservancy.org/events. We would like to express our gratitude to two organizations who have made this possible. The first is the Natural Resources Defense Council, which has been a leader in environmental stewardship and specifically efforts to curb the use of neonicotinoids. They have been an invaluable partner in the Bee Conservancy's advocacy efforts. The second is Villa Maria Earth Garden, whose support has also made this webinar possible tonight. Now I'm going to turn off the chat and hand it over to our host, Rebecca Louie, the Bee Conservancy's Executive Director, to get this program started. Take it away.
Thanks so much, Letta, and thank you everyone for joining us today for this really important topic. As we all know, bees are in crisis with a quarter of the world's 20,000 bee species facing the risk of extinction. This matters because bees pollinate 1 in 3 bites of food that we eat and 80% of the world's flowering plants. One of the factors contributing to bee loss today is chemical pesticides, among them neonicotinoids. So today I'm so excited to introduce a panel of true expert VIBs who are going to talk us through the impact of pesticides on bee biology, how and why neonicotinoids, also known as neonics, came to prominence, the impact neonics have on the environment and humans, and what's being done and what you can do to curb them. We will have a final Q&A session at the end of the presentations, so make sure to drop your questions in the Q&A and upvote your favorites. Our first speaker is Scott Dobrin, an assistant professor of biology at Eckerd Collegium of Natural Sciences. Scott's research focuses on linking molecular and structural changes in the brain with changes in behavior. Using insect models that display complex social behaviors, specifically the honeybee and the bumblebee, his laboratory studies the impact of environmental stressors on learning and memory. Take it away, Scott.
Thank you so much, Rebecca. Let me get my screen shared. Here we go, and hit the button. Wonderful. Does that look good? All right. So I'm so glad to be here today and have a chance to talk to everybody. What I'd like to, before we get started, what we're going to do is actually start on the first slide. So what I want to do is first just set the stage and talk about pesticides. Neonicotinoids are a type of pesticide, but they're an insecticide. Yeah.
Scott, this is Rebecca. I think we might be looking at the wrong screen. We're seeing your background.
Oh no, that's not what we want to do. Let me get
I know that was such a juicy preview, and I know everyone's like ready for the good stuff.
How about that? Does that look right?
Yes. All right.
All right, beautiful. I'm so sorry. Okay. Thank you, Rebecca. And thank you, everybody. I guess we're just having a day of technical problems. But what I wanted to do was just introduce pesticides in general. Pesticides can be broken down in a variety of different categories. And one of the easier ways for us to think about it is based on the target. And so insecticides are an example of a pesticide that are aimed at killing insects, typically small insects, whereas things like herbicides like Roundup are aimed at different plants, fungicides at fungi. We have rodenticides that are going to be aimed at our rodents. And then we even have some more novel, some newer pesticides that are aimed at particular forms of insects, where we're looking at like the larvae, or a larvicide. Unfortunately, understanding the category of pesticide based on the target doesn't really give us a lot of information about it. And so another way to categorize them is based on their chemical structure, or really, and more importantly, based on their mechanism of action. And so looking at insecticides, what we can do is break them down based both on what they look like, which I'm not going to show you, but more importantly, how they're acting. Organophosphates, which are sort of the one of the older pesticides that are out there, as well as carbamates, both are working on a similar synapse. And that's what we see over here. This is what's known as a cholinergic synapse. We have a presynaptic neuron, which is releasing neurotransmitters onto a postsynaptic neuron. And so these are two neurons that are communicating with one another. As neurotransmitters, in this case acetylcholine, travel across the synapse, they bind to receptors, and they're going to activate those neurons. Now, nearby on the cells, we have enzymes that are going to break down that acetylcholine. And this enzyme needs to be active, and it sort of controls, prevents excess stimulation. And so both the organophosphates and the carbamates work by inhibiting this enzyme. When we prevent the activity of this enzyme, a normal release of acetylcholine ends up flooding the synapse, and we have what's known as excitotoxicity. We have excess stimulation of the synapse. Now, we're going to come back to the synapse in a little bit when we talk about neonicotinoids, but we'll see that it's done in a very particular way. Organophosphates, you may have heard of as being incredibly toxic, and that's because while both of these chemicals families can bind to the enzyme, the organophosphates bind irreversibly, and the carbamates can will bind and let go, which makes them a little bit safer. The next classification are known as pyrethroids. Pyrethroids are also going to lead to excess stimulation of a neuron, but here we have to look at a slightly different area of the neuron. Instead of looking at the synapse where two neurons communicate, where one axon terminal talks to the next, we're going to look at the actual axon itself. These are where the electrical impulses, the action potentials are occurring. And if we take a zoomed in look at the axon, we would see that a particular family of proteins known as voltage gated sodium channels are really important in creating action potentials. What pyrethroids do is they prolong the activity of these sodium channels. So again, with a normal stimulation of the neuron actually becomes overstimulated. We actually get more activation, again, leading to excessive stimulation. Now, a modern approach are these insect growth regulators. These are aimed at not really killing the insect, but disrupting its developmental life cycle. And so a lot of us are familiar that insects start off as eggs and then hatch into larvae, which then go into a pupal form for metamorphosis before emerging as adults. What the insect growth regulators do is they either aim at hormones such as juvenile hormone, or they inhibit the creation of chitin, which is important in the exoskeleton of the insect. What's really nice about insect growth regulators is it's aimed at the younger bees, and so, or the younger animals, the younger insects. And so the adult insects are actually thought to not be as affected by them. And it's also thought to be much safer for humans as well. But the reason why we're all here today are to talk about neonicotinoids. And neonicotinoids are a family of insecticides. Imidacloprid is perhaps the most well-known, but there are 7 others. And neonicotinoids work back at our friend, the cholinergic synapse. And so here we see our presynaptic neuron releasing acetylcholine that's going to be nice and happily degraded by our enzyme. And what the neonicotinoids do is they act as if they were the acetylcholine. We don't even need activation of that neuron to release normal neurotransmitter. We throw it on ourselves, and then these neonicotinoids bind irreversibly. They activate the receptors, and there's really no letting go. The nice thing about neonicotinoids is that these receptors are they have a different function in insects than they do in humans. And so it's thought that because this function is different, it allows them to be a little bit safer in humans, although more research really needs to be done on that. Neonicotinoids can be applied in a variety of different ways to the plants. They can be sprayed on a plant, they can be applied to a seed or directly injected into a soil. These are systemic pesticides, and so once they get taken up by a plant, they can be, they'll spread throughout the plant, including into the pollen and nectar, where they're then exposed to different insects, as well as our bees. The other sort of side effect is that these pesticides can then stay in the soil and then get stuck into the water as well, which can really be an issue. So what I want to do now is start looking at specifically imidacloprid, as well as some of the other neonicotinoids, but really imidacloprid is the biggest one that people are studying right now. And they're studying it primarily in honeybees, but we'll see some other examples in bumblebees and other native bees. And I've broadly organized some of the changes that have been shown based on a variety of different exposures. One of the frustrating things about the way that bee research is done right now is there's a wide range in the doses that people are using and the duration that people are using. And so I'm going to be very generic in my approach to this, but there's a wide range of effects. And I think that that's the important thing, is we're clearly seeing effects. So the first set of effects have to do with physiological changes. Through some really surprising ways, the bees' sensitivity to sucrose, their ability to detect low levels of sugar in water, has actually changed. And related, but possibly more concerning, is this fact that when you give bees a choice of sugar by itself or imidacloprid containing sugar solutions, they actually prefer the imidacloprid containing solution, suggesting they're actually going to drink a larger volume and maybe expose themselves more than they would have otherwise. We all know that honeybees, as well as other bees, are really dependent on bacteria, both in their gut as well as for honey production. And it turns out that imidacloprid exposure changes the bacterial community that is really tightly regulated in honeybees. And that's been changed in honeybees, and there's some preliminary evidence to suggest in bumblebees as well. And if you treat an entire colony with imidacloprid and then you challenge it with nosema, it seems that the colonies are becoming more susceptible to nosema, a common infection that can really put a colony at risk. And so colony level physiological changes as well. When we look at the brain, and yes, bees have brains, we can see a variety of changes both in the structure as well as the function of the brain. If you look at a category or a group of stingless bees known as osmia, as well as bumblebees, the volume of various brain structures are changing. When we look at the antennal lobe that I have highlighted here, this is an area that's involved in processing odors, and that exposure to imidacloprid actually changes the way the odors are coded in the antennal lobes. And then the mushroom body is an area known for learning and memory. And if you remove the mushroom body from the bee brain and then treat it with imidacloprid, the way that the neurons fire actually changes. We don't know what that means, but there's likely an effect going on there. Also important is looking at the behavioral changes. This was perhaps the most striking thing to me. I started studying what I thought was sublethal doses of pesticides, but for long periods of time. And very quickly, we start seeing changes in motor functions. I saw bees that couldn't fly well. And in the literature, you see postural changes. They have a hard time supporting their body weight. In stingless bees, you see walking changes. Really concerning is there's a variety of studies looking at foraging related changes, including decreases in efficiency, how much nectar and pollen they return with on a foraging bout. Their ability to navigate is actually decreased, and that ever so important waggle dance to communicate information within the colony is also being disrupted. One of the things I do is to study learning and memory is to do it in a classical conditioning paradigm that I have a picture of over here, where we take bees and we restrain them into small tubes, and then we expose them to a novel odor as well as a sugar reward, expecting them to extend their proboscis, this little mouth-like part, in anticipation of the sugar reward, just like Pavlov's dogs. And it turns out that treatment with imidacloprid actually causes a deficit in learning and memory recall in both honeybees and bumblebees. And maybe, the biggest behavior of all is colony collapse disorder. In a study that used high doses of imidacloprid by dissolving it in high fructose corn syrup, 15 out of 16 colonies were actually were shown to have a colony collapse disorder like phenotype. That's what they expressed. There's some problems with that study that maybe it's not the most field realistic, but certainly strong evidence that something is going on with exposure to imidacloprid. And then of course, a lot of the things we've been looking at are in adult bees. The baby bees are probably being affected as well. And so if you see a frame like this, that might be a concern for your brood, and imidacloprid might be the cause, because we've seen brood production decreases, we've seen lifespan decreases, there's a decrease in the size and the rate of growth of wild bumblebees, and the quality of queens, as well as the number of queens that are being produced in bumblebees are also shown to have decreased. So as you can see, imidacloprid certainly is impacting bees in a variety of different ways. And I think we need to start wondering about how we approach this with the environment and how we're going to look at it in the future. So with that, I'll leave it to Rebecca and take it away.
Thanks so much, Scott. That was a lot of information and it's a little troubling. And to help us sort of parse out what we just heard and put it into a much broader context of the impacts that neonicotinoids are having on bees at large, but also our environment, I'm going to turn it over to Dan Rochelle. Dan is the acting director of the Natural Resources Defense Council's Pollinator Initiative, which focuses on protecting our nation's bee populations from the ever-growing threats to their health and existence, in particular, the use of bee toxic pesticides. Rochelle was formerly co-director of the NRDC's Community Fracking Defense Project and an advocate for the cleanup of industrial pollution in the New York region. On to you, Dan.
Well, hi, everyone. So I'm going to attempt to share my screen here. And hopefully everybody can see that. Can you see a big sign saying caution?
Looks good.
Excellent. Yeah, so I am going to talk today more about neonic pesticides and why we're so concerned about them. I think Scott did a really good job of breaking down sort of how they work in bees, the fact that they are neurotoxic, the fact that they permanently bind to nerve sites and overstimulate bee nerves. And essentially, if you've ever seen a bee that has been poisoned with neonics, they begin to sort of shake uncontrollably before becoming paralyzed and dying. But of course, these effects, even at sublethal levels, cause all sorts of other problems. But I wanted to put things in a bigger context, why we're so concerned about these pesticides, not only from a bee perspective, certainly from a bee perspective, but also an ecosystem perspective, a human health perspective, and some of the things that are happening around the country and around the world to address the widespread and wasteful use of these pesticides. So real quick, there are three big concerns that we have as an organization with neonics. The first is their phenomenal insect toxicity. These are among the most insect toxic pesticides ever created. And to give you a sense of scale here, just one neonic treated corn seed can have enough active ingredient to kill 250,000 bees. And we see that neonics have made U.S. agriculture 48 times more harmful to insect life since their introduction in the United States. And if you look at where that toxicity really kicks up, it's in the mid-2000s. And for those folks who know, this is exactly when we see bee colonies in the United States start to die en masse. Before that, beekeepers were losing 10, 15% of their hives. mostly in the winter due to the cold. And this is the point at which those losses jump to 40, 50, 60% a year, happening year round, right? Beekeepers are losing their hives in the summer as well to colony collapse disorder, to other disorders. And it maps really well with neonics. We know that other things are a problem. We know climate change is a problem, habitat loss, but there is nothing that maps as well as the sudden rise in the use of these pesticides that happens at the same time we see those losses. Second problem with neonics is that they're very good at contaminating the whole environment. So as Scott described, they're systemic, which means they're designed to get into the plant, the leaves, the roots, the fruit, the pollen, everything, and essentially make the plant itself toxic to bees, or well, toxic to insects. Their intended target is pests, but it's toxic to all insects. And that means they can be literally painted on crop seeds. And here is a close-up of some neonic treated corn seed. They don't always pick the same color, but they always sort of look like toxic Skittles. And the idea is that the pesticide is supposed to literally be soaked up through the roots of the plant as it's growing. But the problem is only 2 to 5% makes it into that target plant. The other 95+% stays in the soil where that active ingredient persists for years. And anytime it rains, anytime there's lawn watering or irrigation, those neonics are moving through the soil to contaminate new soil. If there are plants in that new soil, wild plants, those plants soak up the pesticides, they become toxic, their pollen, their nectar. If there is a water supply nearby, that water supply becomes polluted. Third problem with neonics is that they are just all over the place. They are the most popular insecticides in the US. Close to 100% of conventional corn is pre-treated with the neonic on the seed before planting. That's 90 to 100 million acres nationwide, something like 5% of the land area of the continental United States. So big area. Soybeans as well, you're talking 50 to 75% pre-treated with the neonic. So, 40 to 50 million acres, and that's just 2 crops. But outside of the agricultural space, neonics are also all over lawns, gardens, golf courses, you name it. And actually in those non-ag landscapes, they're approved at much higher levels per acre. So to give you a sense of scale of use, we do a lot of work in New York. So a lot of these slides are going to be New York specific. But here is the use of neonics in New York. And Scott was talking about imidacloprid. That's the red area here on the New York chart. And notice how there's a good amount of imidacloprid being used in New York, but it's not even the most used neonic in that state, and certainly not nationwide. But to give you a sense of how much neonic use there is, here's a map from 2003 to 2014. I know 2014 is not super recent, but it's the last year that we have accurate data from. And you can see there is just a massive use of this one chemical, not even the most used nationwide. And note too that this is just agricultural use. So this doesn't count the lawns, gardens, and golf courses. A lot of those white spaces would be filled in if it were. But what it does give you a sense is that these pesticides are being used year after year in the same places, meaning that they're building up year after year because most of it remains in the soil and it persists in the soil for years. So you have a buildup of toxicity year over huge areas of the country that, again, are expanding every time it rains. So, we see this in the water. And again, this is a New York specific slide, but it's similar across the US. About half of the surface waters tested across the US test positive for neonics. A lot of these tests that are done only look for imidacloprid. So again, only 1 of 5 major used neonics that are used across the country. So these test results are likely an underestimate of the true neonic levels that are in surface waters across the US. We also find neonics in groundwater, which is scary, and certainly areas like Long Island, where there's high permeability or high communicability between the surface water and the groundwater. And one thing to note too is that we do see neonics in drinking water in places where they test for them. So there was a good study in Iowa City that found neonics in tap water there. We do see them in Long Island as well in the tap water there where they test for them. And note that traditional chlorination does not remove neonics from water. Only advanced filtration does, but very few water systems use that type of filtration. So I promised I would talk about something other than bees. It is worth noting, though, that there is a wealth of science that connects neonics to the losses of honeybee colonies. But honeybees are not the only bees. They're not even native to this country. There are 4,000 species of wild bees that are, and they are similarly being hammered by these pesticides. But beyond bee populations, neonics are broadly toxic to a whole range of insects. And we see across the world insect populations going down in the last several decades. And the example I give to people is something entomologists have told me. It's sort of the windscreen effect. If you remember in the 90s, if you were around then in August, driving through a rural area, you'd have to turn on your windshield wipers, whether it was raining or not, just to get all the bugs off your windshield. Does anybody remember that? When's the last time you had to do that? And that's sort of the one place where people do see, aside from honeybees, the impact of these pesticides on the environment. They are literally hollowing it out from the bottom up. And that, unsurprisingly, has impacts on other species. So birds eat insects. And so if we have fewer bugs, we have fewer birds. And there's good studies in the United States and also in Europe making that connection. Also worth noting that neonics are directly toxic to birds and other vertebrate animals. So one neonic treated seed has enough active ingredient to kill a small songbird, but even at levels that don't kill those birds, they can cause all sorts of sublethal impacts again. Issues with the ability to migrate and navigate, so neurological issues, issues with being able to put on weight right before migration, which is important, issues with reproduction, immune system harms. So even if the neonics don't kill birds outright, which they certainly can and do, they can cause a loss of that bird eventually or a loss of the population. Beyond birds, we know neonics are similarly harmful to fish. So similar issue here. A good study out of Japan found that, these folks were studying zooplankton, and these are zooplankton for those unfamiliar. They were looking at a sustainable fishery, and this is one of the few studies where we have a data set from before the world before neonics, because right now neonics are sort of everywhere. But they were studying the zooplankton in this sustainable fishery, otherwise sustainable fishery, and saw that in the mid-90s, that population hit a wall. And unsurprisingly, the populations of fish that ate the zooplankton also hit a wall. And the study authors later linked that to the use of neonics or the introduction of neonics in nearby agriculture. This is right when they were first approved. And it's worth noting that we see similar levels in water across the United States that were later found in that lake. Things get a little scarier with studies linking neonics to birth defects in white-tailed deer. This is a study that grew out of weird things that hunters were seeing in the wild with the deer that they caught. And lo and behold, they did a study and did link it to the use of neonics and found that there were higher rates of all sorts of impacts, including deformed jaws, decreased organ weights, and increased mortality for deer fawns that were prenatally exposed to neonics or exposed in the womb. There is some human health research as well. So CDC monitoring finds that about half the American population is exposed to a neonic on any given day, meaning that is literally in the bodies of half the American population on any given day. People become exposed through contaminated food and water, as well as direct contact. And while we don't have a ton of human health science at this point, because neonics are relatively new, the science that we have links them prenatal exposures, so exposures in the womb, to autism-like symptoms, birth defects to the heart and brain. Animal studies link them to all sorts of other issues, so thinning areas of key areas of the brain, like the cortex, also reproductive harms, And we actually just got a trio of new studies linking neonics to non-neurological conditions, which is kind of interesting. Decreased testosterone and impacts or associations with alteration of insulin levels and weight obesity. So I know I'm already over time, but very quickly across the world, a number of folks are taking action on neonics. So the European Union has already banned the 3 major neonics in heavy use there, and the ones that are used on seeds in particular. Quebec and Ontario are largely phasing out of neonic treated seeds for corn and soybean. In the United States, and I know Kathy will talk more about this in her presentation, EPA is unlikely to do much about neonic use. They came out with an opinion recently that would essentially continue to allow widespread use, and we think they're going to approve or finalize that opinion this fall. And we think that EPA has been underestimating human health risks as well as the risks to the natural world. States, therefore, are taking action. New Jersey and Maine recently banned non-ag uses of neonics, and other states have taken action to restrict them to certified applicators. So, landscapers and other folks, although that doesn't cut down on most of the use statewide. Just want to highlight this bill for those that are living in New York. There are bills across the country now on neonics. There's one in Connecticut. There is this bill in New York. So if you're in New York, we have 2 weeks left in the session to pass this bill. It is a prohibition on sort of the, I call them the dumb uses of neonics, but the high cost, low benefit. These are the widespread use of these pesticides, but that either don't provide benefits for users, like the neonic treated corn, soybean, and wheat seeds, or are easily replaced with safer alternatives like the non-agricultural uses. The bill would eliminate about 80 to 90% of the outdoor neonic use in New York. It is an exceptionally smart, tailored, and wonderful bill. Again, there are 2 weeks left in the session to pass it. We do think it's possible, but we got to get across the finish line. So if you live in New York, Rebecca is going to have a QR code for you to scan so that you can call your legislators. We need all the support for the bill that we can. And I should also note there is a bill in California that would ban non-agricultural neonic uses. The bill number there is AB 2142. And again, also bills in Connecticut and across the country. In general, if you want to support these pieces of legislation, the best thing to do is to call your legislators. Believe me, it is so impactful. They care. If you call your state representatives, they will take note. We also have petitions. That link is to a petition in New York. But again, right now in New York, the thing to do is to call your representatives. Writing letters to the editor, to your local paper are also impactful. And of course, getting local groups involved, social media support, and so many other ways to get involved with these issues. But I will stop my share there because I know I'm already way over time, and I will pass it to Kathy.
So Dan, thanks so much. I'd love to introduce Kathy. That was a sobering insight into where we are. Those maps were a little devastating. And I think Kathy is a perfect person to sort of give us the context of how we actually got here today. And so just so we understand who Kathy is. She is an environmental contaminant specialist for the U.S. Fish and Wildlife Service. And as the lead pesticides expert in her region, Kathy coordinates with the Endangered Species Program on all activities regarding pesticides and endangered species. She has worked as a technical lead for litigation-driven Endangered Species Act Section 7 consultation with the Environmental Protection Agency regarding the registration of pesticides and their effects on endangered species. And for those of you watching at home or at the office, we know there's a lot going on. So if you have questions, again, use that Q&A function while we have Dan, Kathy, and Scott here to answer your questions. Upvote your favorites, and I'm going to turn it over to Kathy.
Hi, thanks, Rebecca. So is my slideshow up?
Yeah,
oh,
looks good.
Awesome. So I want to thank Scott and Dan for setting the stage for me just to really get into some of the issues around neonicotinoids and also just pesticides in general. So when I use the word pesticide, I mean, just like what Scott was saying, the whole umbrella, the herbicides, the pesticides, the larvicides, and most of the insecticides, because those are the ones that have been proven so far to be the most lethal. And so neonicotinoid insecticides are actually pretty new on the market. And they came on the market because EPA began to look for alternatives to organophosphates and carbamates. And I also want to mention pyrethroids. And so the organophosphates and the carbamates are your, your malathion, your diazinon chlorpyrifos. Carbamates, the best known one is carbaryl. And these products are being phased out because of their toxicity. And I kind of want to note here that when we talk about the pyrethroids, those aren't they're fairly new as well when you look at the whole history of pesticide use. But they were brought in as an alternative product because of the toxicity of the organophosphates and carbamates. So to follow technology and some of the mistakes that we don't ever refer back to history. The OPs and the carbamates were brought into the market to replace the everlasting, very persistent organochlorines, DDT, the most notorious one, which was an ecological disaster. So in the mid-2000s, as Dan said, neonicotinoid use increased rapidly, and it just really went up. And that had a lot to do with the way that it was put on the seed and having it be seed coated. It's thought by some that makes it a safer application process because you don't have drift of the non-targets. But as we've seen, that's not true. It's a systemic insecticide, which means it's going to move in the environment. It's going to move through the pollinators, through the soil and into the aquatic environment. And this is just kind of an overview, kind of backing up what Scott and Dan both said, that imidacloprid was the first registered neonicotinoid. That was in 1994. So that makes it really new on the scene because we've been using pesticides for a long time. More of the neonicotinoids came up through the market in a very short period of time. When they hit the market, they went off like gangbusters. And it is the most widely used insecticide in the world. Across no measure, it's the most used one. And the popularity, again, was because of this innovative idea of putting it as a seed coating, because then it won't hit a non-target per se. But again, being systemic, it's problematic. So I kind of want to discuss a little bit just a major problem with pesticides in general. And this is the registration process. And for years, decades, even before I came on with the Fish and Wildlife Service, which was over 20 years ago, EPA does not consult with the United States Fish and Wildlife Service under the ESA. And under the ESA, which is the Endangered Species Act, Section 7 says any federal agency or entity that has an action that will affect a listed species must consult with either the U.S. Fish and Wildlife Service or National Marine Fisheries Service, the two agencies that implement the Endangered Species Act. Well, EPA believed that they had the in-house knowledge. So this promulgation happened, this agreement occurred where EPA said, well, we have toxicologists, we have this in-house knowledge, we can do our own Section 7, and they never consulted. And I will say this, that if they would have consulted with the service, these adverse effects that these pesticides now in our environment have had, we would have found them, we would have seen them, and the regulatory leg that kind of rules over the pesticide use is the legal document called the label. And you have to follow the label. And that's one of the issues that Fish and Wildlife Service has with EPA is that the labeling is not protective for listed species. For the most part, it's not protective for anything other than doing the mission that it's set forth to do and to go towards taking out pests for a certain crop. So this lack of consultation with us, with the Fish and Wildlife Service and with NIMS, led to national consultations on multiple species. And this has been a long process. Back in 2007, the Center of Biological Diversity sued EPA for not consulting with the services on the use of pesticides in red-legged frog habitat. That was the first big lawsuit, and that one won. And then after that, the Center of Biological Diversity started pursuing more lawsuits, and they could not lose. And this was an excellent win for those of us who are very concerned about the chemicals going out in the environment. So what happened during this litigation is they wanted us to backtrack and to kind of evaluate the pesticides that are already out there. Most of them are the commonly used pesticides that we've been talking about, the insecticides in particular, diazinon, malathion, and carbaryl. And a lot of these products aren't necessarily, I don't want to say they're not being used, but they're not being used as much. So pyrethroids were also added. At that time, neonicotinoids were not on this list. And that consultation took on all kinds of different faces. And then it led to a large national consultation where we're looking at all species and all the pesticides, which sounds really great. But unfortunately, that umbrella effect is now we're using surrogates for susceptibility, for adverse effects. And I should probably make this clear. When we say an adverse effect, when we say likely to adversely affect, that leads, we're talking about avoiding the extinction of the species. So the Fish and Wildlife Service, in their mission to do these consultations, we would look at the potential of these chemicals leading to the extinction of a species. This is coming after the bald eagle, the picture that I have of this marvelous bird, the national bird, was going in declines because of eggshell thinning, directly linked to DDT. Now, I'm not saying if we would have consulted on DDT before it was registered, would we have known this? Maybe we would have, maybe we wouldn't. But taking the services, taking the entities that are mission to look after conserving the nation's lands and habitats and wildlife species, we should have been part of that conversation. So moving forward to 2013, EPA, Department of Interior, Department of Agriculture, and Department of Commerce came together and decided we need to figure out what we're doing because there's all these lawsuits now. And in 2013, the Academy put out a report saying that the services and EPA need to come together. And then this year, in January, EPA announced that they will be working with the services, and for any new registered product, any new active ingredient. So also the registration is the active ingredient in a product, not necessarily the whole pesticide, all of its mixtures and how it's introduced into the environment, that they were going to consult with the Fish and Wildlife Service. And this is also, to be known, this is litigation driven and not a very popular stance that the EPA is taking from the registering applicants. So moving away from that whole background of, maybe if EPA had done what they were supposed to do, and I don't want to point any fingers at EPA necessarily, but if we were using these laws and these processes that we have in place to protect our environment, maybe we wouldn't have some of these problems, or they would be negated a little bit. And some of the neonicotinoids use that we took very big concern on was on the use on national lands. So even though the National Wildlife Refuge System is part of the U.S. Fish and Wildlife Service, they still need to do an intra-service consultation when they start using different products. Since neonicotinoids are registered, they don't have to do that consultation because that federal nexus has already been avoided, has already been overlooked. So what they do is basically do plans, a pesticide use plan and pesticide use reporting. And the Ecological Services Division branch of the Fish and Wildlife Service, which is where I work, we evaluate the pesticide use on our national wildlife refuges, and we're allowed to kind of do a little bit of consulting there. And our national wildlife refuge systems, and I don't know if this is common knowledge, but Fish and Wildlife Service is the largest land manager for the people of the United States. The mission of these refuges is to protect biodiversity and keep the integrity of conservation of species and habitat. In some essences, even preserve that. And that's part of what this refuge system was set up for. If you look at history, one of the first refuges set up for brown pelican. Numerous refuges here in California, where I'm located, are managed for endangered butterflies. Elaine's Meadow Mark at Antioch Dunes, Smith's Blue at Salinas River National Wildlife Refuge. So there, when we have those type of species on our refuge, we're like no chemical use. One little hiccup, one little move that could take out just one could be very hazardous to the whole population of the species, because some of these butterflies are they live in these pockets, and that's the only place they exist. So with encouragement from the Ecological Services, the Wildlife Refuge System decided to not use neonicotinoids on their lands. And some of the refuges rely on the money of growing crops on their lands. And so they were using some of these neonicotinoids. And we were just like, you know, we can't get behind that. We know that it's bad for wildlife, it's bad for pollinators, and that's our role. We can't be okay with that. So they pledged to not use it. And as the neonicotinoids seemed like they were going out, the administration withdrew the prohibition of the use on refuges. All the work that we did to make sure that no refuges were using them, the administration for years withdrew that prohibition. And conservation groups have challenged it left and right. And to no avail. Like Dan said, they're still going to be used quite a bit. And I just have a couple more slides that I just want to talk about the schedule of review for neonicotinoid pesticides. So there's an open docket, and you can find this all on the EPA website that will tell you about the decision-making process and when these decisions are supposed to come through and what these decisions will be. Again, it looks like that EPA is going to keep their current policy and how they have neonicotinoids registered, and that they're not going to be putting up any more boundaries on it, if you will. When I say that, it's they have a lot of information out there regarding pollinators and wildlife, but they're not putting any restrictions on its use. And so I'm just going to skip over this. And so, like, if you go to the EPA's website, you'll see, you can find all of those. I thought about putting the tables up, but it'd be just a little too much. And there's a lot of information out there on neonicotinoids. But for the future of neonics, we know they're bad. And that's something that has to be really addressed. There's a lot of evidence for scientists and policymakers to use to make responsible decisions. We've shown that it affects all pollinator species and almost any species that it comes in contact with. Each state can adopt more stringent restrictions, and that's the recommendation. A state by state does not have to do with the federal. In California, we have the Cal EPA, which can put stronger restrictions on the use of a pesticide, stronger than what, and we do have our limitations our restrictions that we put on it are stronger and more conservative, if you will, towards wildlife and the environment than US EPA. And I think one of the biggest problems is that these restrictions are constantly, and these bans are constantly being challenged. Even in the EU, the first area that really put in the bans, they're being heavily challenged, and some of their decisions to not use certain neonicotinoids have been overturned, and I can't see where that's changing anytime in the near future. So thank you for your time, and thanks, Rebecca.
Kathy, thanks so much. And thank you all, Dan, Kathy, Scott, for your incredible insights and context today. We have 2 questions in the chat, and they're sort of the perfect way to close out this conversation today. The first one is from Dr. Ruiz, which is a question for Dan, and I think maybe Scott as well. As a researcher myself who works with farmers and farm services in the country with a focus on farm workers exposed to the environment, what are the direct physical and wellness effects to farmers? What can I do as someone interested in researching bees in my state of Connecticut?
So, the answer there is we don't know yet, because there hasn't been enough science on the impacts of these pesticides to human health. We do have poisoning reports from EPA, so we know that people poisoned with neonics can die. There's actually been a suicide with using neonics. We know that you experience neurological issues, muscle tremors, memory loss. But as that trio of studies I mentioned during the human health slide in my presentation, there are all sorts of impacts that we don't know about or that we're just learning about now. So again, the CDC monitors people's urine across the United States every year for a variety of toxins. And in the last survey, they found about half the Americans that they surveyed had neonics or neonic breakdown products in their urine. And it's just now that people are looking at, hey, what about those people versus other people that didn't have neonics in their bodies? What are the differences? And they're realizing that there are impacts to testosterone levels in people. And again, associations with the level of insulin in people's bodies, also with weight issues, obesity, or actually weight loss in some circumstances, depending on the neonics. So there's all sorts of impacts that we don't yet know about. The ones that we're most concerned about, though, are to the youngest people. So people in, young children, as well as fetuses in the womb, or pregnant mothers that are exposed. Those are the biggest concerns because these are neurotoxic. We do have these receptor sites in sensitive areas of our nervous system. So exposure at a young age can be exceptionally damaging to people's developmental health. And again, there's that study showing the connection between neonic exposures and birth defects of the developing heart and brain. And it's those exposures that we're most worried about.
And as far as getting interested in research and trying to get involved in bee research, the bee community is really, really engaging. I would say just go out and talk to people. There's a variety of citizen science efforts that you can be involved in, talking to beekeepers or beekeeping groups, talking to local, going to the colleges that have bee researchers and seeing what projects they have. Oftentimes they're looking for different people. And then depending on your skill set, you could do it yourself. There's a really amazing group called, amazing website called scientificbeekeeping.com that it's just an individual doing small scale research on his own. And it's he's a scientist, but he does really amazing stuff. So you can do it yourself. There's a lot of different options out there.
Scott, thanks so much for that. And the whole do-it-yourself ethos is pretty exciting because, we do, as citizens, as gardeners, as community members, have a lot of say in how things go. So, Sandra asks in the Q&A, what can young people, specifically ages 8 through 17, do to learn, speak out, and advocate for change in the use of toxic pesticides? Kathy, do you have any thoughts on that?
Yeah, there's so much to do because it's such an overwhelming topic. But one of the best things you can do is to start in your own community, having conversations with young people, with your parents. Notice to see what they're using in their yards and look for alternatives. There's always an alternative than using a chemical. And then moving on from that, maybe inviting a guest speaker to come to your school. Reach out to people like me and Rebecca, Scott, and Dan. Reach out to people like us who can maybe point you in the direction of people that can come to your school and talk. And then if you have an environmental club in your school or in your area, join that and be very vocal, but also prepare your young person to be to get a lot of blowback from it. It can be a very hard road to take because a lot of people are really stuck in the mode of using chemicals. That chemicals is better living through chemistry concept is still alive and well. But the biggest thing is just inform yourself and just be really aware of what's going on and the chemicals that are used in your area, and then maybe have like a block party. Have the internet is a wonderful thing. You can create chat groups or start, or if you don't have a club at your college, your junior college or your university or your high school, maybe start one and just start by just asking a lot of questions and just being curious and finding out. But know that it's the people who ask the questions that go out on these limbs are the ones that are making the differences. So my thumbs up to anybody who wants to do it. I've been doing it for a long time. It can be hard, but just start at home, start in your own backyard, and then move it out from there.
Scott, any things you'd like to add to that?
I think that Kathy made a lot of good points. The only thing I'd like to add is just not being scared. It's not so much with pesticides, but this idea that we need to get rid of all of the bugs around us and that they're out to get us. I think getting over that will maybe allow us to appreciate the fact that these little insects do really neat things and that, they're alive and really interesting as well. So, yeah.
That's a great point. And I'd just like to share, Dan had to leave literally because he is engaging the Senate in New York right now to help pass this Birds and Bees Protection Act. But you will see on the screen, there are some QR codes for you to engage with some active campaigns. And as Dan mentioned, because we're so close to New York State, to the actual voting process, calling right now is a bit more powerful than signing a petition. And from the point of view of the Bee Conservancy, first of all, I appreciate Kathy and Scott and Dan for sharing your thoughts with us today. And I love this idea of like getting in the community and engaging and doing the work, because what we think about a lot with our organization is, what are the different intersections in, right? Like maybe you don't garden and maybe you're not interested in insects, but maybe getting back to children, like there are sports fields and parks nearby. Right. So if you're running around in the grass and, you have systemic pesticides there that are then entering like the entire soil system, and you're kicking up dirt, and, there are lots of ways to think about it. People think about food justice, food security a lot. And if you are part of a program that's about community resilience, this is directly related. And so, using the creativity about what you care about and connecting it somewhere down the stream to where there's an intersection to take action, it's usually easy to find. So, with that said, again, thank you so much to our panelists today. Thank you for the great questions that we had, and for everyone who joined us and was patient with our technological moment that happened in the beginning. But we hope to see you soon for another BTV program. And until then, be well. Thank you so much.